morning, everyone. Welcome to today's session on the AI revolution in biopharma. It's my pleasure to introduce a wonderful set of panelists moderated by David Reese, who is the EVP Research and Development, Amgen. I'll let him kick it off. Thank you all for coming.
Well, thank you, and welcome everyone for what we hope will be a terrific session on the artificial intelligence and machine learning revolution in biopharma. I'll ask the panelists, and we've got a really fantastic group this morning to introduce themselves in a moment. But what I wanted to do is sort of set the stage by kicking it off with a simple observation. You know, everyone up here is doing the sort of work we're doing. There's so much excitement that you can hear the sirens in the background. You know, all of us believe that we're what I like to call a hinge moment in our industry, a moment where things will qualitatively change, and the drug discovery and drug development process will be different in 5 and 8 and 10 years from what's really come from decades before that. And what is driving that hinge moment or that change is a union of biotechnology and technology. And that's what we're really going to, perhaps we'll pause just a moment. And that's what we will talk about here today, how that union is actually coming together to create something new. So why don't we go ahead and have our panelists introduce themselves, give a little bit about your background, because some of you on stage are coming from backgrounds that may be a little less familiar to those in our audience. Mike, you want to kick things off?
Thanks, Dave. Good morning, everyone. My name is Mike Nally. I'm the CEO of Generative Biomedicines. I joined the organization 2 years ago after having spent 18 years at Merck. In many respects, I became frustrated that I think there are two seminal challenges that face the industry. One is that, unfortunately, R&D productivity for the industry has been going down for decades consecutively. And then secondly, I think in some ways somewhat interrelated, that has led to bad pricing practices for the industry. And as I reflected, the only way to start to solve those sort of challenges is through the union of science and technology. And I believe, we're on the precipice, as Dave noted, of understanding biology in a way that the human mind alone couldn't understand it through the aid of technology. And that's what's so exciting to me about this panel, this point in time. We think, at Generate, that the world is going to become, in biology, much more engineerable. And, that will migrate from this artisanal craft to something that can be productionized in a way that we've never been able to do so. And so we're excited to share more about it with you today.
Great.
Wonderful. Thank you for the invitation. I'm Mark DeCristo, one of the co-founders and CEO of Big Hat Biosciences. My background has been at the intersection of bio and tech for about 20 years. I was a computer science undergrad, but I got a PhD in biochemistry and bounced around from places like the Broad Institute and Google's AI division before founding Big Hat in 2019. And we're focused on combining advances in synthetic biology and machine learning to make better antibody drugs faster. And what that really means for us and what we think about in the world is just the key things that are changing are knowing that we can make molecules quickly and measure their properties. And then we have new technologies that let us learn from them. And that allows us to go really quickly through designs of molecules to try to make better drugs quickly.
Great. Alex?
Hi, I'm Alex. I'm the VP of Technology at Unlearn.ai, and we work on AI-powered clinical trials. So everyone else works on getting more drugs to trials, and we are innovating in the clinical trial space and speeding up that process. I, and actually a lot of people in the company, have a diverse set of backgrounds. I'm a physicist that loves machine learning and biology. And we have a group of people that are looking to innovate and want to change up how we think about machine learning, take advantage of all the great data and new tools that are out there, and see what new problems we can apply this to. And we chose clinical trial spaces, but this whole panel is tackling the whole process everywhere.
Great, thanks. And Jacob?
Hey everybody, thanks for coming. I'm Jacob. I'm a co-founder and CEO of Therapeutic, and I think, echoing what everyone else said and what Alex touched on, I think we're at this moment where the data is, finally there within biotechnology and drug development research to unleash the power of AI and accelerate what we're doing. And for me, I come at it from a small molecule chemistry side, from the drugs you take in a pill. And we were talking last night, I briefly go down memory lane because it's illustrative, like, did my PhD and postdoc making small molecules myself, figuring out ways to make them more efficiently. And probably in that whole journey, I don't know, I made like a few thousand molecules max, and it took like 8 years or whatever. And then, when we went out to look for academic jobs, my friends were going down to Merck, and they came back and were like, oh my goodness, we got these automated robots. They can set up thousands of reactions. We can do it in like a couple days what you used to do in a year. And I was like, oh, wow, that's pretty incredible. And I set off in my own academic career then to figure out, like, how could we even further miniaturize that, drive the numbers way up? And so the core technology that's at Terray, we built in my academic lab before I left that lab to found Terray, and we're able to measure tens of millions and hundreds of millions of molecules in a precise, iterative fashion at tremendous scale. And I think that type of custom-built data for the information age allows us to bring therapeutics forward much faster by pairing it with the AI piece. And so we're right up here in LA and delighted to be here.
Great, thank you. And Jacob, I'm going to come back to you in just a moment to kick us off with questions. But I thought I would also make sure that we're all level set here in understanding what the drug discovery and development process actually entails. And this is a model that has been in place now for really half a century or a little bit more. It starts with some insights, usually incomplete, into human biology and disease pathogenesis. And ultimately, that zeroes in on the identification of a pathway and ultimately the target of a drug, a target or target, so usually a single target. At that point, you then make a choice. How can you interdict that target? Typically, it's one of two things. You're either trying to turn it off or, less frequently, turn it on. And so you then embark on molecular engineering to develop the right sort of what we call modality. Might be a small molecule, like an aspirin. It might be a large molecule, like an antibody, or now gene therapy, small interfering RNAs, cellular therapy, or a host of other modalities. There's an absolute profusion of modalities that we now have in the toolkit. Ultimately, you select a clinical candidate. That goes through various preclinical studies, including toxicology, and is then ready to advance to the clinic. Clinical development is roughly divided into early phase and late phase. Early phase focuses on safety and an initial proof of concept that the drug's actually doing what you hope it would do. And then later phase development is really designed to prove that this is an advance on what is currently available and will help move the needle in terms of treatment of disease. Then there's the post-marketing setting. After you have regulatory approval, that's actually when we often learn the most and go back to the laboratory and start iterating. Now that process I just described in 2 minutes takes 10 to 14 years on average and costs somewhere between $1 and $3 billion to prosecute. And we succeed about 8 to 10% of the time. So we clearly need to do better, and everyone here is coming at this in a little different angle to help us do better. In fact, at Amgen, when I became the head of research and development about 5 years ago, I instituted 3 strategic imperatives. And those imperatives were very simple. Number one, reduce the cycle time, meaning we have to do better than that 10 to 14 year process. So how do we do that? We'll talk about that a lot in the coming minutes. 2, increase the success rates. We have to do better than 8 to 10% of the molecules going into the clinic, making it across the finish line for regulatory approval and marketing. And then third, what we won't talk about a lot today was increasing access and use. Now that actually may be more than half the battle, getting payers and healthcare systems around the world to actually use the drug in patients who can really benefit from it. Well, all of you come to this drug discovery and development process at a little different node. And so what I'd like to do is start with an explanation of your technology. I think we'd like to hear about what kinds of data, and you began to touch on this, Jacob, are now available that we didn't have even a few years ago, and how you're using that to really address some of these questions of cycle time, success rates, and beyond. So Jacob, why don't you start?
Thanks. Yeah, I think for us, so within that spectrum of the drug development, we work on the aspirins of the world, trying to find a way to make them, like, we got teed up, faster, more effective, higher success rate. So the first two strategic pillars is what we work on. Much shorter cycle time, higher success rate. And so the core technology that drives Terray is this ability to make very large collections of candidate drug molecules, little small molecules that might work, and we make millions and millions of these now, tens and hundreds of millions of these, and then we're able to put them in these little microarray chips, these little chips the size of a nickel, which is the core technology, and we put 32 million per chip, and we can measure every single molecule in there in under 5 minutes. And so that's a huge transformation from when I was doing my PhD and postdoc, where I was testing like 10 molecules. And so over the last year, we've actually collected 1 billion unique measurements between small molecules and protein targets. And to those sequence that was described, a key part, when I think of this sort of three legs of the drug development stool, it's got to work, it's got to be safe, and then it's got to get where it needs to go. And so because we have this huge upfront chemical map of all the molecules against all of the targets we run, we actually can start off sort of way ahead on both that it works and that it's safe because it's selective. And so we start with selectivity and potency up front. And then the key piece is that the cycle time is accelerated too, where we're able to go from very big data, build machine learning and other computation models, and come back around and test like 2 to 10 million new molecules in under a month. And so that's a scale and cycle time that just didn't exist before our technology. And so internally we work on it to treat immunology diseases, and then we partner with other companies to tackle their challenges in a variety of other areas.
Yeah, that's fantastic. And Jacob raises an incredibly important point, which is the scale of what we're doing is now increasing by logs. We're using another technology to hunt for drug prototypes called DNA-encoded libraries. Typically, as Jacob has described, for small molecule drug discovery, you start with a library that may have up to a couple million compounds and find, for example, fragments that bind to your target of interest, and then you begin doing iterative cycles of chemistry to refine that molecule. Now, using a technology where we've got 1 billion fragments in a test tube about the size of my little finger, we put the target of interest on what's essentially a fish hook, dip it inside that test tube and find out what binds to it. And each one of those fragments has a DNA barcode that encodes what its identity is. You then just simply sequence the DNA, takes a few minutes literally, and you identify that fragment, and then you start from there. So this notion of scale is critically important. Well, Mike, scale is something that you're thinking about as well. And full disclosure here, Mike, our companies have a collaboration, and we're thrilled to be doing what we're doing together. But why don't you talk about the technology and what your thoughts are?
So Generate was founded 4 years ago to try and understand the generalizable principles that govern protein function. As we looked at biology, really, proteins are the workhorses of biology. About 95% of all biological functions are driven by proteins. And so if you could ultimately understand these principles that govern function, you could program novel protein-based therapeutics with desired attributes. And so as we started the company, and Gevorg, our chief technology officer and co-founders in the audience, proteins was a perfect place to start in biology. Oftentimes data sets are all over the place, but in the protein domain, you had the sequencing revolution, and companies like Illumina give us 180 million amino acid sequences across every species on the planet. And then you had the protein data bank, which had about 200,000 high-quality, high-resolution structures of proteins. And by learning on that data, we were ultimately able to start to understand interrelationships between protein sequence and protein structure. And as we set up the company in Boston, what we were trying to add from day one was a third leg to that stool and understand function. And so we have spent almost the entirety of our time miniaturizing our experiments. We've been working on automating our wet labs. So we take a computational-based approach, but it's fully integrated with a laboratory on the back end to try and generate functional data at a scale that we've never seen before. And I know there are a lot of tech investors here. When we talk size, we're not talking anywhere near the size of data sets that you're well accustomed to. These large language models blow away the scale of data in biology. But with thousands, if not millions, of data points, we're starting to actually understand these principles quite well, and that's allowing us now to program molecules at a speed, a pace, and a scale that was never possible before from a drug perspective. And again, I think this moment in time does signal this transitional point from an artisanal drug discovery process, where a genius or a team of geniuses have come up with an idea to come up with the next molecule, to a period where biology is becoming engineerable. And whenever complex fields become engineerable, you see industrial revolutions occur. And I think we're at the early stages of that in biology. There's a long way to go. By no means have we cracked the code, but I think you're going to start to see advances at a pace and a scale that we've never seen before.
Yeah, another incredibly important couple themes there, which is this era of engineerability, which is now upon us, and the notion that drug discovery, which used to be empiric to a great degree and iterative, multiple cycles. You generate a molecule, you see what it does in a series of experiments, then you refine it, you go back and start again. That's what drives us to a 10 to 14 year cycle time. Can we engineer away a number of those iterative cycles? And how are we doing that? Combining a wet lab, the traditional wet lab, with dry labs, so in silico design. And that is something, Mark, of course, that you've been thinking about a lot in your career. So I'd love to get your thoughts here.
That's a perfect tee up for more about Big Hat. I mean, that's exactly what we're focused on. At Big Hat, we're really trying to combine two huge advances. There's one in AI that's letting us design molecules better. And there's a whole world of that, from AlphaFold to these large language models. And we leverage all of that internally. But you're ultimately also critically dependent on data in bio, and experimentation needs to get higher scale and faster. And so we're really on this high speed trajectory. We have a synthetic biology wet lab, and what that really means is new technologies to let us write DNA, create molecules outside of cells, and then we test those really quickly. And this combination of AI and synthetic biology lets us make thousands of molecules each week, individually characterize them, and then learn from that data so that the next Monday we make new designs and drive this cycle that used to take kind of 4 to 8 weeks is now every week at Big Hat, making molecules, seeing what they do, experimenting on cells, checking that they're in trajectory for actually moving the needle therapeutically, and going faster and faster at that. And what ultimately I think we're building, and it goes back to this question of access and use, ultimately, I think the drug development is shifting, as Mike was saying, from kind of a bespoke artisanal process to something that's engineered. And we think of ourselves at Big Hat as building a scalable drug development machine. And the goal here is to capture all the best practices of how to make molecules, analyze them, learn from them in one reusable platform, and that we could improve that over time and use it to take many more shots on goal. Because ultimately, it's the shots on goal that let us pick the most successful therapeutics and advance them into patients.
Fantastic. So I think you get a sense of the excitement and the fact that we really are at this hinge moment. Well, Alex, I'll ask you to comment in a minute, because you're at the other end of the spectrum here, and you're now working on the clinical trials portion of drug discovery and development. For me, as a head of research and development, this is by far and away the single largest budget item that I deal with. Late-stage clinical development accounts for far more than 50% of the total cost of developing a new drug. Oftentimes, it can be 70 or 80% of that cost, depending on the scale of trials that are required. Well, to tee up, Alex, I'd like you to ponder one thing. The randomized clinical trial, which is sort of the gold standard, is exactly 75 years old. So this year, the first randomized clinical trial was published in 1948 in the British Medical Journal, and it was a trial of streptomycin, a new antibiotic that was being tested for tuberculosis, which was untreatable at the time. And there was a famous statistician who'd been agitating to do randomized trials, and he couldn't convince anyone to do it because they felt that it was unethical. Well, there was an extreme shortage of streptomycin. So because of that, they said, well, it's ethical to actually randomize people to getting the antibiotic or getting standard of care. What was standard of care? Bed rest for 6 months and good luck. And the trial had a randomization, it had blinding, it had central review of x-rays, all of the things that we now incorporate in the modern clinical trial. And when you think about it, not much has changed in that 3/4 of a century about how we do these trials. But again, we feel we're on the precipice of a new era and a new way of doing clinical experimentation. And so, Alex, I'd love to get your take on this.
Yeah, Unlearn was crazy enough to try to take on this thing that stood for 75 years, and we've had success. And so fundamentally, the clinical trial hasn't changed. If you want to test an Alzheimer's drug, you find 2,000 people to test it on. 1,000 of them get assigned to the actual treatment. 1,000 of them just sit there and do nothing. And that hasn't changed for 75 years. Well, what if instead you could make a digital twin of somebody that comes into the trial, and then you could, for everyone that gets the treatment, you could have a digital twin that doesn't get the treatment, and that would allow a deeper insight into what happens to somebody during a trial. And now this completely changes what you can do. You could now maybe sign up 1,000 people in total, and 800 of them get assigned to the treatment, and only 200 have to be in the control. And that's what we focused on at Unlearn. And you could say, like, why now? Why has nobody tried to do anything in this space for a long time? Well, fundamentally, this randomization and blinding is super powerful. It is a tough thing to beat. I mean, this controls for a lot of the noise during a trial, and it was a big game changer. It did pretty well. So why take that on? But now the type of data that's out there, so we come in at the later stage. We come in like, we get clinical trial data. This is like doctor comes in and assesses you. And the amount of data that's out there has, just blown up in the last few years. And so the data's there, and there's new models, right? Fundamentally, you need generative AI models to make sense of this, right? We have to, given some initial input about a person, you have to generate, the future of like what this person will do under different conditions. And, that technology just wasn't there until recently. And so, yeah, that's why now is the perfect time to take this on.
And it's hard to overstate the importance of this for our industry. 90% of trials that are done in biopharma don't end on time. And so there's a staggering cost associated with that. 50% of the sites that sign up to participate in the trial never enroll a single patient. So you've gone, obviously, to the wrong place. So we need these kinds of data sets now. We need predictive algorithms to tell us how that trial is going to enroll, where it's going to enroll, and how to do it quickly.
Hey, may I add one other thing?
Yeah, please.
Yeah, I think the pandemic opened up our eyes in a major way as an industry, right? So we're all talking about how technology is coming into the fore. The reality is, to solve this longer cycle time issue within the industry, you're going to need all of these different technologies, interconnected, certainly. But you're also going to need to work differently with stakeholders. We're all going to need a different mindset. Patient safety has to be paramount as we develop new drugs. At the same time, many people in the industry didn't believe coming up with a COVID vaccine within a year was possible.
Yeah. And
what we've seen is now more people being immunized against, COVID than any other vaccine that's ever been used in the history of the planet, almost. So I mean, in many respects, there are ways we're going to need to work differently, not only on the technology side, but also with regulators, with health systems, and other stakeholders to get these medicines to the patients that need them
Yeah,
fast.
Mike, absolutely critical point. And what I like to say is that the pandemic taught us the art of the possible. You used the word possible. It's the art of the possible. How can we go from idea to an approved vaccine in roughly a year? And why do we not systematize that for drug development going forward? Here's a thought. In the last 3 years, about 10 times as many people died from cardiovascular disease as died of COVID. Where's the sense of urgency? Where are the emergency committees? Why are we not bringing that kind of paradigm to bear on the single largest public health problem that we face? Well, let me ask another question to all of our panelists now, and we've got a large number of investors that are tuned in, and the question is very simple, which is why now? What's different now compared to a few years ago? And then what do you think we need to look at in 5 years, 10 years, 15 years? What sort of milestone should an investor be thinking about? What should they be watching in terms of progress? And I'll start a little backwards this time at the far end of the spectrum with Alex.
Yeah, I mean, so why now? I mean, fundamentally, we're all doing AI, and AI is powered by data, right? So the amount of data that has become available in the last couple of years, it just blows away anything that was there before. And then okay, so you have a pile of data. What do you do next? Well, you need the right model, the right framework, the right way to think about it and process this. And this is where generative AI, everything that has been going on that you see with ChatGPT, all those models have been sitting there. We're all super excited using similar things, and that has unlocked new capabilities of new types of questions you can ask with that data. And and then finally, you need the right use case, the right context of use. And I think that's something that why has no one else tackled clinical trials? It's because we have to go and partner with the FDA and say, hey, look, we have this cool new generative model, and we come in with a new statistical method to analyze this. And so we can, that's how we marry and fit this into the clinical trial space.
Yeah, you also bring up, I think, an important point, and this is sometimes a point of friction between the worlds of tech and biotech. Biopharma. We live in a highly regulated environment. We like to say we're the second most regulated industry on earth, next only to nuclear power. And sometimes when I have conversations with folks from the Valley, experiments are proposed, and I say, that's wonderful. It's a great idea, it would answer the question, but we're actually dealing with human beings here, and there's certain things that we can do and other things that we can't do, and so how do you sort of bring those two worlds together and have the conversation? Well, Jacob, let me turn to you and ask the why now question.
I think I'll give the same answer, just slightly different, because Alex is 100% right that it's that intersection of data and the tools. I often say that drug development is not just an algorithm problem, it's a data problem, but we're benefiting from the tools that have been developed in data sets that are large, precise, and iterable, if that's a word. But I think you see that, where, AI has transformed our lives, and you see it. It's in data sets like, Netflix viewing patterns, or Amazon purchasing patterns, or Facebook click patterns, and these are huge data sets, right? Truly big data, like Mike said, huge data sets. They're very precise. You know exactly how many minutes of exactly how many shows everybody watched. There's no ambiguity. And then they iterate. You build a model, and our models are generally not perfect the first time, even for simpler tasks. But then they iterate. 100 million people watch stuff again the next week, and your model can evolve and get better and better. And so same with ChatGPT, as we talked about, right? That came on the back of multiple different language data sets that built and built and built. And you see it even in AlphaFold, right? AlphaFold is built on top of crystal structures, which the data set grew and grew and grew.
Let me just jump in there, because I was going to ask, why don't you explain to everyone what AlphaFold is and the notion of in silico protein structure prediction, because everyone here may not be familiar with what we're talking about.
I'd be happy to, although I feel a little bit
chagrined
doing it in the presence of Mike and
well.
Mark as the small molecule guy. But essentially, these models are training on one set of data to then predict an outcome. And so AlphaFold is one that trained on a growing body of protein structure data to the point that when the data set was large enough, it can now make its own generative predictions about structures it hasn't seen, proteins it doesn't have fundamental base data for, and be extremely accurate and give you a starting point now for the rest of your work with a prediction that would have never existed before. I think as we talked about offline, like there were structures that we would have loved to measure, we would have loved to see, but experimentally people tried for years and couldn't get it. And now by teaching the computer what a subset looks like, it can actually predict that one that maybe you couldn't measure, you couldn't get to.
Yeah, and I'll ask or two other folks here to comment in just a minute, but the context here is that this protein structure prediction was actually a holy grail in biology from the day after Watson and Crick's paper was published. Could you go from the primary sequence of a gene to understanding the three-dimensional structure of that protein and now how that protein is interacting with other proteins? And that, we're in the middle of a revolution. Mike, why don't I have you? You and Mark have spent a huge amount of time thinking about this, making use of the technology. Love to get your thoughts.
Yeah, I think one of the great things about the space right now is we're all building on each other's innovations. We've incorporated structured prediction into all of our standard workflows. So this is the AlphaFold capability, and by building on each other's fundamental insights, we're all pushing the field forward at a pace that's truly exciting. For us, this question of why now, in some ways is simple. Our core model that we've trained on would have taken 100 years to train a decade ago. The computing power wasn't there in order to actually do this. And so, these tools have never been available to biologists. And I think the other thing that's really, really important, when I joined Generate 2 years ago, I remember sitting down with Gevorg and sitting there saying, how many people are experts at the intersection of machine learning and biology? And there were about 120 in the world.
Yeah. You got half of them are sitting on stage.
Exactly. And so you're in this space where, to solve these problems, it requires multidisciplinary expertise. Our universities were not training scientists in this way. Nowhere in the world were scientists being trained in this way. And so for us, a huge part of this problem, but you're seeing, a supply and demand dynamic occur where these people are oftentimes recruited by tech companies. That's who ultimately is our probably biggest competition for talent. But increasingly, we're seeing them come into industry to try and fulfill a sense of purpose to try and improve human life.
Yeah, I think you bring out an incredibly important point, which is, for us as well, we recruit people now, data scientists. They can come from anything from theoretical physics to finance. They have to have certain things in their toolkit, and the value proposition is, hey, do you want to try to cure some really bad diseases? So, and that's a pool of people that 10 years ago we never would have thought about. Well, Mark, love to get your thoughts on the protein structure prediction revolution and the why now question.
Yeah, those are both great questions. Actually, I studied and did my PhD on protein structure prediction 20 years ago, and this was a field. Yeah, even then it was unclear. I think no one thought that it was coming, that there would be a solution to this problem. And I think it's been hugely impactful for the community, in particular for our companies here, because the best thing about AlphaFold, in my experience, has been that it's a tangible, usable piece of software that most people in biopharma could experience and see the improvement over what they could do before. And at the moment it was available, the skepticism of the potential for AI shifted immediately to enthusiasm. And so AlphaFold itself, in many ways, is a critically important scientific advance, but it's just culturally shifted the narrative in a way that's benefited, I think, everyone here, and certainly will encourage more and more investment in this space. And so, of course, it's a huge breakthrough and all that, but I think the implications on the future of drug development have been huge culturally. And I think that's against this backdrop that is really a great time to be in this space and investing here. People have mentioned we're moving from a data poor to a data rich environment. That's phenomenal. We're also moving from slow and big experiments to kind of fast and targeted experiments. And I couldn't emphasize the importance of that point anymore. The large data sets that we generate in biology are sort of table stakes for everyone. We all can access them. They're hugely enabling. But what you really need to create, and I think every company here has talked about their data generation strategy, what's really unlocking drug development on top of this background of like AlphaFold is being able to build and construct your own data sets that are specific to the drug development challenges you're going after so that you can generate the custom data you need for that problem. And so that's why we emphasize this integration of sort of synthetic biology and the AI tech, because you need a way to generate data in a targeted fashion. And ultimately, that's all driving this shift that everyone is talking about here of moving from a kind of science-first drug discovery, a history where you notice some weird mold growing and then you discover penicillin. I mean, that's great. It's a huge breakthrough, but it's not exactly easy to put an OKR in to do something like that. And then shifting to a world where you're really engineering something. You know the targets, you know the disease, you know what it does and why. You know so much more about what you're trying to accomplish that these problems are started becoming ones of, okay, well, I know what I need to do. How do I make something that actually does that? And I think that's why so many people are so excited about the AI revolution and why it's happening now.
That's fantastic. And to give you a tangible example, at Amgen, we were trying to get a protein of interest, a potential disease target, and I've got teams of the best protein engineers, protein chemists in the world. Half a dozen attempts over a couple years. For various technical reasons, we couldn't get it. When DeepMind deposited data, in the public domain, several 100,000 protein structures, there it sat. It wasn't perfect, which is another point. Doesn't have to be perfect, but it was a starting point. We used that, and in 2 months, we had the protein in hand, and we're doing experiments in the wet lab. So think about that. 0 for 6 over 2 years versus a 2-month cycle. That will take years off your cycle time, and it will improve your success rates because you're starting at a much more intelligent position. Well, you've heard a lot of the excitement today. Let me ask as a sort of final question here, come back to this notion that I alluded to. What should the folks in the audience be looking for in the next 5, 10 years, for each of you? What are important milestones? Mark, maybe I'll start with you. If I'm an investor and I'm thinking about when do I jump in, what are the sort of milestones in your area that they should look at?
Oh, that's a great question. My feeling is like we're past those milestones. Like these have happened. We've just talked
Right
about
now.
them. Like now is the time to get in. And the argument for it, I think, is pretty clear. Like this is an enormously huge market. Therapeutics, antibodies alone are $300 billion a year in sales. The whole drug development market is even bigger. We have this new technologies, which we're all sort of have different flavors of. They're ultimately pointing at exactly the things that David brought up as the key to changing drug development, right? Higher success rates, faster, and higher quality. And ultimately, because of the nature of drug development, on a 10 to 15-year timescale, kind of all diseases are in play again because of the nature of patents. So everything that's on the market today is potentially going to be succeeded by better therapeutics in that 15-year timescale. And so I think what you see is this kind of great reshuffling coming, where you're going to have science-first companies becoming more engineering-first companies. And that market is sort of all in play, and the earliest sort of nucleation points are starting to appear today in some of the companies that are represented here.
Yeah, and you bring up another important point that is top of mind for us, which it used to be that when a drug lost exclusivity, went off patent protection, that was sort of the end game. Now, more and more, that drug will be technically or technologically obsolescent because of drugs coming behind long before it actually reaches that end of patent exclusivity. So something that we need to keep in mind as we think about staging investments. Well, Jacob?
Yeah, I mean, I think we'll come full circle because I think you probably nailed it when you started setting your goals, right? I think the milestones I agree, the time is now. It's happened. But the milestones you'll see are the shrinking of cycle time, the increase in success rate, and it'll come through larger scale experimentation at a reduced cost, accelerated speed, deeper into the funnel. Like historically, to get through that, it's a funnel, described as a funnel for a reason. Everything triages at each step because it gets progressively more expensive each step in drug discovery that you move down the line. And so you got to drop things earlier and earlier to focus your dollars as you go later. And I think you'll see the impact of AI in that more projects can go further, have more data, better decision making, and that'll play out in improved success rates, faster cycle times. And I think you see that in a company our size, historically, you would have worked on 1 or 2 things. Like there's no way you'd tackle more than that. And then with our technology, we tackled 9 problems in our first year as our first wave, and that just never would have happened before.
Fantastic. Alex?
Yeah, I mean, I agree. Now's the time to invest. You got to go all in. And why are we all so excited? Well, it's fundamentally, like we were saying, like there's new tools that are unlocking just new things that no one thought was possible. And importantly, this whole cycle time is so slow, right? We're going from something that could take 15 years. It's going to be significantly sped up. And I think something people don't quite realize is there's a huge compounding effect on that. People today that are working on these problems are going to learn so much more than they could have in a whole lifetime before in just a few years. And then once you start to learn that quickly, like all of a sudden we're going to start disrupting other adjacent areas that we're not even touching right now. And so that's why it's very easy to see that, well, you could have 10 times as many drugs in 10 years. And the people that can innovate faster will be able to take a larger share of that.
Fantastic. Mike, bring us home.
Yeah, I want to pick up on the thread that Jacob touched on. We haven't touched much on scalability. Again, computers are inherently scalable. And I used to work for a 72,000 person company that would start 10 to 15 programs a year. I now work for a 200-person company that does the same. You're seeing productivity in ways that has never been seen in biotechnology. And so for us, I think there's a lot of reason for optimism that this is going to fundamentally change the way every drug is made over the course of the next 5 to 10 years. And when you couple that with the simple fact that, it's one thing to say, the technology is starting us on a path. These programs are now going into the clinic. Our first program will go into the clinic in mid-year this year, and it was started on February 11th of last year. So within 14 months, you're into human trials, and that was something that used to take...
Years ahead of schedule.
Exactly. And so, this is a really exciting time for the industry. It's an exciting time for humanity because, everyone in this audience, in their families and loved ones, are affected by some sort of disease. And the more we can bring these tools to ultimately address those diseases, I think we're going to alleviate a lot of pain and suffering and help people live longer, healthier lives.
Well, I think that's a perfect note to end on. We're all here because we believe passionately that ultimately it's delivering drugs to patients that change the practice of medicine, change the natural history of disease. I hope you had a flavor of the excitement that we've got in terms of how this is going to be done in a qualitatively different fashion. I want to thank all of our panelists again. I'm sure they'll be happy to stay around and answer questions, but I think we'll conclude our session there. I really want to thank all of you for your time and attention.